So under what conditions would slow growth be adaptive, right? So in these shorter experiments, we're not looking at macroevolutionary transitions. We assume that cells are dividing asymmetrically already. They're not going to divide more asymmetrically. And Ostriococcus is not going to suddenly make a germline, right? So given that you can't change major parts of your life history, what are your options? Well, your options are, so here we have, this is just my little cartoon. If you're going to reproduce slowly, so you have a long arrow here, you make little damage, and then you can make offspring cells that are brand spanking new, ready to go. Whereas if you're going to reproduce really quickly, you'll build up damage and pass it on to offspring. So when might this be the better strategy? In a case where you're being selected to grow fast, right? It's obviously the better strategy if all you're doing is persisting. If you want to grow faster, either you have to have offspring that can tolerate this damage, you need to invest more in repair so that you end up with no damage. You need to slow down damage, so you need to increase growth without damaging yourself. That's another option. Or if all of those things fail, you can slow down growth, right? And I call this option, this slowing down growth, the prodigal son hypothesis. And I have a much better illustration. So here's what happens. You have an environment by this dotted line here, and this environment is initially awesome. This is the environment, the organism's preferred environment. It may not be the best environment. It may not be the best environment in theory, but it's the environment in which the organism lives. So it is the best environment for it in reality. So it's realized niche that way. The realized niche then improves. So cell division rates can go up. A lot of traits will change to accommodate this. Photosynthetic rates are going to change to accommodate this. All sorts of other metabolic traits are going to change. Then you have three options. You can ramp it up. So you can grow even faster, and this is what we all expect, right? If we select for rapid growth and give things more food, they'll grow even faster. Keith Richards takes this approach. You tolerate more damage in the more permissive environment and just keep trucking, right? And that's not unrealistic. If the environment is better, perhaps you can tolerate more damage. Perhaps it's less important that every component of the cell be functioning perfectly, right? And this is really true. Like the 20-year-old Keith Richards, after his night of partying, probably had to sleep on the floor. The current Keith Richards probably sleeps in a 5-star hotel. His environment is much more permissive, right? So any damage he's accumulating is probably not quite as horrible as it would be in a worse environment where he still had to sleep on a concrete floor in his own vomit. Okay, then you have the David Bowie approach, which is that you hold steady. This is sort of like you change a lot of things, but by and large, you keep up the same level of doing everything that you've always done. You just do it differently. Given 2016, I have to find a new approach there. And then you have the Paul McCartney approach, which is that you just just dial it right back, right? And all of these work, but they may not all be evolutionarily accessible to organisms. So first thing you do before you invest like 4 years of graduate student time in anything is you do a simulation. So the first thing I did was a simulation. And what I did was I just simulated the organisms. We're not going to go through the whole simulation. But the important thing was I selected them for rapid growth. And then they inherited damage from their parents. They then damaged themselves more based on how fast they were growing, or damaged themselves less if they were growing less quickly. So you have a parent with a growth rate, you inherit that growth rate with some mutation, you inherit your parent's damage times 1/2, right? And then based on your growth rate, your damage either goes up or down, and based on how much you're investing in growth, you can invest more or less in repair. And then we select on both reproduction, which is basically faster you're growing, the higher your reproductive rate, but also on viability, and viability is based on damage, not growth rate. And this is the mean growth rate that evolves. What I'm showing you is what happens after a few, I think 10,000 generations. This is the mean growth rate that evolved in the ancestral environment. So in the old environment, the maximum possible growth rate is 1. The growth rate that evolves is about here. If you have some damage, that's what we expected. And in the new environment, the maximum possible growth rate is 2. And you can see that in some cases, a growth rate of almost 2 does evolve. And that's where you basically just inherit your parents' growth rate. But if offspring growth rate has anything to do with damage, so as you get towards this end of the axis, this is the contribution of offspring growth rate to damage. You can see as soon as the offspring growth rate affects cellular damage at all, it's really difficult to evolve a higher growth rate. In some cases, even though the environment improves drastically, growth rates don't go up at all. There we go. So that was a simulation. And I went through all the different strategies. So here's the maximum growth rate that evolves in a reference scenario, environmental enrichment, where you can't change how damage and growth are related. You can't invest more in repair. You just take today's organisms and tomorrow's world. You see that it's hard to evolve rapid growth rate if there's a damage associated with increased metabolism. If you can tolerate more damage, you can evolve higher growth rates. So this dot has moved up. If you can make repair cheap, and that may be possible if there are more resources in the environment, you may be able to devote more to repair without taking anything away from growth. So that's cheap repair. And then you can slow damage. So that's the idea that in a permissive environment, damage is less important. Anyway, so the take home from that experiment was that this idea of accumulated damage is certainly sufficient to limit the ability of winners to sustain high growth rates indefinitely in improved environments. The caveat here is in this simulation, you're assuming they start in a good environment anyway, right? So they're not starting in a horrible, horrible environment and then going to a tolerable environment. They're starting in an environment that they're perfectly happy in, and then an environment that improves. So if they're going to increase their growth rate in the improved environment, they need to evolve some strategy for dealing with the damage or a way to tolerate it. So you can think there are many phenotypic solutions to this, there are many ways to do it, but if you can't actually get around it, the only thing left to do is to reverse that change and lower your growth rate. So, and I call it prodigal son because one, I don't know if you guys have noticed that all evolutionary phenomena are named after Alice in Wonderland characters who are now out of characters. So I moved on to the Bible, and I think that would keep us busy for some time. And two, it gave me no end of pleasure to name an evolutionary hypothesis after a Bible story. But for those of you who haven't brushed up on your Bible stories for Lent, the prodigal son story is a story of a son who becomes suddenly rich, takes his money, runs away, lives fast and hard, and then depletes his resources and has to go back home and return to a slower, return to the ancestral way of life. So this is a case where living fast and hard is in itself what causes you to go back to something slow. There is no prodigal daughter hypothesis yet, but Jeff Morris has made promises, so I'm waiting. So basically, the world of SineadCollinsTalk assumes this. It assumes that as the environment gets better, as time marches on, that they're going to be able to keep on increasing their growth rates. What we see in experiments with some things like Chlamydomonas, which is a freshwater algae, and Trichodesmium, is that they can maintain the plastic response, but they do so by changing other traits. So a lot of their regulatory traits change. For example, to do this, Chlamydomonas stops regulating its carbon concentrating mechanism, and so it becomes unable to grow at normal levels of CO2, so it becomes an obligate high CO2 organism. Trichodesmium does something very strange to its nitrogen uptake regulation. So they're able to sustain those growth rates by making other changes, other regulatory changes. Ostriococcus has to reverse traits. It reverses almost all of the plastic responses that it sees that it does in response to high CO2. The only one it doesn't reverse is making lots and lots of external polymers, which I guess makes sense. You have to jettison the carbon somehow. And there could be something to this. Ostriococcus has a minimal genome. It may not have the evolutionary potential to change all the other things that we need changing. It's also a very small cell. It doesn't have anywhere to put damaged bits, if that makes any sense. Chlamydomonas is a huge cell relative to Ostriococcus. So I can come up with all sorts of just so stories. But the point is, if we want to know who's going to win in these enriched environments, we need some way of predicting which of these trajectories different types of organisms are going to see and are going to have access to. This is, of course, the winning trajectory. But I have yet to find an experiment that follows that trajectory. So how are we testing it? So what I've shown you is a bunch of results that were sort of consistent with this idea, or that led me to this idea, and then I've shown you two models about asymmetric cell division and my model, right? So I've shown you nothing real. But Rasmus to the rescue. So this is a student who's halfway through his PhD with me taking a selfie. This is what fun he does. And he's testing the proteolyson hypothesis with Chlamydomonas. And what we've done is we've taken Chlamydomonas reinhardtii, we've taken 4 different genotypes of it, and we're growing them in CO2 enriched environments and, of course, ambient environments. So here's our enriched environment, here's our control. And we're growing them either in bottlenecked populations or in large populations. A bottleneck population means that when we transfer the cells, we transfer so few cells that natural selection is unable to act. So these bottleneck populations are going to evolve, but natural selection will be completely ineffective. So they can't adapt, they can only evolve. Whereas these control populations are large, natural selection should be very effective in those populations. So we're trying to ask, what does natural selection see? And in addition to that, in each of these treatments, we also have lineages where we're adding exogenous ROS. So because the hypothesis is explicitly about oxidative damage, we also have treatments where we don't increase the CO2 or increase the growth rate, we just increase the oxidative damage to try to account for that. So we're here at the end, but we haven't done any of these for A or B yet. So he's currently measuring growth rates and whatnot, and it sounds like he's just being lazy, but it takes about a month to get all the growth rates measured. It's a very big experiment. There's a few hundred cultures there. So, but these are extinction rates. So this is what I can show you. We were wrong, as usual. So here's the entire selection experiment, 150 days. I think it went to 2. We went to we ended up at 300. This is the first 200 days of the experiment. These are the accumulated number of extinctions, which is interesting. So if you have a small population and you're at ambient CO2, that is the worst thing that can happen. Almost all of those populations went extinct. Because of the short term, high CO2 is protective. This is still the short term for Chlamydomonas. If you're at high CO2 and we add ROS and we take away natural selection, you still go extinct, but there's a huge protective effect of high CO2. If you have big populations, if natural selection can act, your extinction rates are very low. And if you have high CO2, your extinction rates are very low indeed. And in fact, there were no extinction rates in the high CO2 with no added ROS with big populations. So we certainly see that there's an effective selection, which we expect. So a small population go extinct at higher rates. There's a protective effect of high CO2 early on when most of these extinctions are happening. See, they tend to even off. And there were no more extinctions after day 150, which is, so we just stopped plotting. And it's bad to have ROS. So that all worked. But annoyingly, we didn't seem to see that it was very bad in the long term to be in this high CO2 environment, even with small populations. So we weren't seeing the meltdown we expected here. But what we did see in the surviving populations, so what I'm showing you here is, these are growth rates. So Chlamydomonas can divide every 8 hours if you grow it in 24 hours of light and really nutrient-rich media, which is basically what we were seeing in this experiment. We wanted them to grow fast. That's the whole premise of the experiment, so we did. So these are the ambient CO2 selection lines grown at ambient CO2 and high CO2. This is a really rapid growth rate. These are just different strains. And these are the high CO2 selection lines grown at ambient CO2 and high CO2. And what you can see is actually the high CO2 selection lines that have survived have decreased their growth rate. So we have actually seen that came through as we were expecting, which was nice. So they've taken this approach. Oops, I went backwards. Sorry, we're not going to do it again. So I'm going to tie this up, because I'm not over time yet. So this is the stuff from this talk, which is what I showed you at the beginning of the talk is one example of how trait reversion is widespread in improved or multi-driver environments. I didn't show you other examples, but I have, like I said, 16 years of them at this point. And I put or multi-driver there because the experiment that I'm just finishing, which has just been submitted, we used 96 different environments, so multi-driver environments with 1 in 8 drivers, and we found the same thing, that in the majority of these multi-driver environments, even the ones that weren't improved, they were just complex. we saw massive trait reversions. So it was really interesting. And this is consistent. So what I'm showing you is data that's consistent with selection against producing damaged offspring. This is prodigal son dynamics. And what we need to figure out to sort of apply this to understanding what future communities are going to look like is which beasties are able to take the Keith Richards approach. These will be the winners. And depending on how you feel about Keith Richards, that may sort of rub you different ways. This is other stuff my lab is doing currently, and I'm putting this up here because I'm going to be here for another little while and I would like people to bother me. So one of the things my lab works on is we look at how heritable epigenetic effects affect genetic adaptation. We've even done some dreaded epigenomics and have lived to tell about it, barely. We look at this multi-driver question, how do the number, identity, and intensity of drivers affect short and long-term responses? I'm especially interested in pulling apart sort of how the number of drivers versus, so how the sheer number of things changing affects evolution and plastic responses versus how the identity of what the particular things are affects it. And the concrete way of putting that question is how accurate do our scenarios have to be when we're trying to project forward. And then finally what I'm working on currently is what are the joint roles of plasticity and diversity in determining evolutionary potential. We actually know a lot about plasticity, we know a lot about diversity, but no one has been brave or stupid enough to put them head to head in one giant experiment. So Tatiana Reinerson and I are doing that with a bunch of Antarctic diatoms right now. I'm done. I thought these are the two graduate students who produced all the data you saw in this experiment. This is my lab drinking chlorella mojitos. And this is the lab, I think, this winter, and as of when I get back, the lab will be just me and Rasmus, who wasn't even here for this picture yet.